Good day. I'm Anne Beatriz J. Valencia, and together with my groupmates, I'm Manuel Canlas, Beverly Constantino, J.V. Galan, and Marvin Gian Villanueva. We're going to tackle all about the hazards, risks, and protection from a biosafety level 2 microbiology laboratory. Here's the list of topics in our agenda for our report. First, one hazard and associated risk in a microbiology laboratory will be identified. The materials for our makeshift personal protective equipment will be discussed together with their respective counterparts with the standard PPE. Our photos wearing our makeshift PPE will be shown as well as a short video demonstrating our non PPE, its advantages and disadvantages. Several ideas will be discussed with regards to how we can protect ourselves in the laboratory setting. And the last part of the report will target to describe the characteristics of a good microbiologist from our point of view. To start, the microbiology laboratory presents the hazard of exposure to infectious agents or biohazards. Biohazards are biological substances that may present a health risk to humans. The use of bacteria, viruses, blood, tissue, and or bodily fluids in the lab can lead to potential biological hazards. These materials can all carry disease or hazardous allergens which could put the lab team at risk. The effects of the disease and allergens can be immediate or take significant time to manifest, demonstrating the importance that all members of the lab team are given sufficient protection, even if the dangers are not yet known. Diseases carried by humans and animals used in research can be transmitted by the team, who then may become carriers. This means that biological hazards could prove to be a massive risk for not only the lab professionals working with the materials, but anyone they come into contact with outside of work.
For our group's makeshift PPE, the materials used were the following. The face shield, this is in replacement of the face protection equipment such as goggles and splatter guards. A surgical face mask, this replaces the expertly fitted N95 mask in the laboratory. The windbreaker and raincoat pants replaces the protective laboratory coats and gowns. The non-sterile latex gloves is in replacement of the sterile high-quality latex gloves used in the laboratory, and shoes wrapped in plastic is in replacement of the shoes sealed with a disposable covering in the laboratory.
So, pictured on this slide is the group wearing the makeshift PPE, and for this slide would be the group wearing makeshift PPE in style.
The first part of the PPE is the head protection, and it is composed of the face shield and the face mask. The advantage of using a face shield is that it is widely available, especially now, and it does the purpose of protecting the handler from potential splashes and contaminated fluid. However, its disadvantage is that it only covers a majority of the face. In biosafety level 2, it is recommended that the face protection covers until the neck of the handler. The next protective equipment is a face mask. The advantage of using the common surgical face mask is that it is widely available and it protects the handler from a majority of microorganisms. However, due to the generic fitting, it is possible for it to not fit properly, endangering the handler. In biosafety level 2 facilities, it is recommended that the handler use properly fitted N95 masks as it protects the handler to a wider range of aerosolized microorganisms.
Next part is the protective clothing for the torso, which is a windbreaker. One advantage of this over the normal gown required as part of the PPE used in the Biosafety Level 2 microbiology laboratory is that a majority of it is waterproof. Therefore, it is somewhat effective in protecting the handler from microbial contamination. However, some parts of the jacket are not entirely waterproof, such as parts made of cotton, like the cuffs or the pockets. Its susceptibility to water could easily result in microbial growth and contamination. It is also disadvantageous that it only covers the torso. In actual PPEs,
The body protection is a gown that covers both the torso and the legs, mitigating the chance of contamination and the transition from jacket to pants.
Now, let's talk about gloves. Latex gloves are similar to what are being used in the biosafety level facilities. They are effective in protecting the handler from getting contaminated while in contact with contaminated surfaces. However, latex gloves have varying levels of quality, and it depends on the brand. The latex gloves used in the laboratory settings are certified to be durable and free from microscopic pores.
Similar to the jacket, these pants are waterproof, so it protects the handler from potential splashes and possible microbial growth or contamination. However, the pockets in the pants could potentially be contaminated, especially if it is made of a different material that is not waterproof. Additionally, potential contamination could occur near the waist, in the area where the pants transition into the jacket.
Next is the protective equipment used for the feet. The feet are also important to be protected during laboratory works since they also have the capability of spreading microorganisms in public areas when not properly covered in the lab. Polyethylene is commonly used for laboratory grade shoe covers. Because of this, we may be able to use common plastics as substitute. This makeshift shoe cover may be achieved by wrapping the whole shoe in plastic by tying the material close, as done by other members of the group, or by using rubber bands to do so, which can be observed here. Since this is only a makeshift shoe cover, there are advantages and disadvantages with the techniques mentioned. The advantage is that the use of plastic to cover closed-toe shoes is important because it protects the shoes of the handler. Protection of the shoes ensures that the handler does not spread the microorganisms to public areas. The disadvantage is that the use of plastic bags or wrap may not provide an effective seal compared to laboratory grade shoe covers. The shoes could possibly be contaminated, spreading the microorganism into public areas. That's all for feet protection.
So, at this point, let's discuss the difference between hazards and risks. So, a hazard is something or any source that can cause potential harm or adverse health effects on someone. A risk, on the other hand, is the chance or probability that a person will be harmed or experience an adverse health effect when exposed to a hazard. Now, to contextualize in the laboratory setting, as mentioned earlier, when working in a microbiology laboratory, the hazard is usually biohazards or infectious agents or pathogens. And the risk in this case would be getting sick or infected after being exposed to that hazard. So the risk of getting sick or infected is decreased when wearing our PPE, and it is increased when we are not wearing our PPE.
Agents, conditions, and activities that are in a laboratory can bring on hazards and risks such as what my group may have reported, but worry not because these can be managed using RAM. Recognize hazards, assess risk, minimize risk, and prepare for emergencies. As already described by another groupmate, these hazards, may it be chemical, physical, and more, other than these, must be recognized. Risks associated with this should then be assessed. With the use of unbiased, accurate information from reliable sources, such as experienced researchers and or safety experts, determine the risk of present physical and chemical hazards, then rate and evaluate them by their impact to personal safety, resources, work performance, property damage, and reputation. Assessing risk in relation to a specific experiment a scientist is about to do leads to appropriate strategies for the next step, which is minimizing risk. Approaches that can be used to lessen the impact of hazards are, but not limited to, PPEs, administrative controls, engineering controls, substitution, and elimination, arranged in increasing effectiveness as presented in the hierarchy of controls from the National Institute for Occupational Safety and Health. Last but definitely not the least, scientists must still prepare for emergencies. Despite recognizing hazards, assessing associated risks, minimizing them, There will always be potential accidents and errors that might occur while doing laboratory work. Being prepared is critical for mitigating effects of the danger that may come along. Additionally, there exist standard microbiology lab practices and safety rules and established laboratory biosafety levels that a scientist can follow to protect laboratory personnel as well as the surrounding environment and the community.
So, how do we describe a good microbiologist? In general, a good microbiologist should exhibit traits essential for any scientist to succeed in their field of interest. Because of this, a good microbiologist should be objective, analytical, knowledgeable, collaborative, and thorough. Being objective and analytical is critical for any microbiologist when conducting studies, as this trait pushes them to abandon bias and look into the study with a fact driven mindset and explanation for certain events. Additionally, being a critical thinker allows a microbiologist to cross examine findings with available knowledge, as well as identify possible errors that could be committed during studies and laboratory works and prepare for them. Collaboration is also important among microbiologists as this promotes healthy discussions regarding relevant topics that may lead to expansion or correction of previously acquired knowledge. Good microbiologists should also be thorough, both in conducting laboratory works and implementing research. Being thorough in the laboratory as a microbiologist calls for being careful and meticulous with every aspect of the experiment. This includes using appropriate safety equipment and clothing or PPE, practicing good hygiene, taking note of safety equipment and first aid kit availability, among others. Lastly, a good microbiologist is thorough with the research being done by being knowledgeable of the different glasswares and lab equipment, as well as safety equipment to be used. Along with this, the knowledge of the microorganisms to be handled or are applicable for the biosafety level of the establishment to be used is also important. That is all.